Boys are back in town, the boys are back in town, and we're back discussing in this video RNA. So RNA is very similar to DNA, except there is some major differences. So RNA, contrary to DNA, is single-stranded. It has a different pentose sugar and different nitrogenous base. So it does have that phosphate group attached to a pentose sugar, but the pentose sugar with RNA is ribose. So DNA, pentose sugar is deoxyribose. RNA, the pentose sugar is ribose. So ribose, RNA, deoxyribose, DNA. But pentose sugar, once again, with RNA is ribose, and also the nitrogenous bases do differ with RNA. So for RNA, the nitrogenous bases are adenine, guanine, cytosine, and uracil. So the uracil takes the place of the thymine from the DNA. But once again, the nitrogenous bases of RNA are adenine, guanine, cytosine, and uracil. So A, G, C, and U. So RNA, they are single-stranded, so it's like one complete side of the ladder. So DNA, there were two sides of the ladder that connect. RNA is just one single strand of the ladder here. So the complementary bases with RNA are adenine and uracil, and the guanine and cytosine are still going to be the complementary bases. So instead of A and T binding together, the nitrogenous bases with DNA with RNA, it's going to be A and U, so the adenine and uracil. And the guanine and cytosine, it's still going to be the G and the C. But the single-stranded here, so it's going to have, let's say, pentose sugar, the ribose, the phosphate group, and then poking out here, we'll have one of those nitrogenous bases. So let's say it's an adenine in this occasion. So that, as we recall, the RNA takes information from the DNA, pretty much splits off into two. So the DNA is in the nucleus, boom, transcription, we got the RNA. RNA travels outside the nucleus, and in order to function properly, in order to produce that protein, it needs to bind back up actually to its other side. So in this case with the adenine, the RNA, it would bind to a uracil pentose sugar and allow the protein to function properly. So here's how it goes. Boom, DNA in the nucleus, take a strand off. Let's say it's a pentose sugar, obviously the ribose with the RNA. This is an RNA. This is the nucleus. We're outside the nucleus now. And our nitrogenous base, let's say, is adenine. So boom, we got the adenine. The complementary base would be the uracil. If this was guanine, the complementary base would still be cytosine. And then it obviously forms the protein outside of that nucleus there. So the DNA is double-stranded. RNA is just a single strand there. DNA, it's double-stranded. It has deoxyribose as the pentose sugar, and the nitrogenous bases are adenine, guanine, cytosine, and thymine. Whereas with RNA, still attached, there's still going to be a pentose sugar, except the pentose sugar is ribose. Still attached to the phosphate group, and there is a nitrogenous base, except this time it's adenine, guanine, cytosine, and uracil. So very similar, except there are some major differences between DNA and RNA. And just as a reminder, DNA to RNA is transcription, RNA to protein production, producing the protein outside the nucleus, that's translation there. So it really is a beautiful thing. The DNA gives the genetic information, the directions for production of protein to the RNA. RNA says, all right, I'm going to take this outside the nucleus. RNA is like, all right, let's make a protein now. We got all the information, all the directions. We just need to put it into action. Then boom, RNA translates to a protein. Proteins control pretty much everything that happens in the body, and all this happens on a microscopic level in such a quick amount of time. It's a truly beautiful thing. Anatomy is in the human body. It's wonderful. We don't have to do anything. We just got to sit back, relax, and keep it at homeostasis.